Thank you for this opportunity to talk with you guys and this good presentation. I hope you enjoy it. And today I plan to explain the reprogramming plant stress physiology based on the reactive carbonyl pathways and the glutathione S-transferase nodes as a synthetic control modules. Here is my outline. So first of all, I want to introduce myself and then a little bit information about plant stress physiology and reactive carbonyl species and glutathione S-transferases and how synthetic control modules related with the plant stress physiology and how can help this knowledge to build our future. So I'm actually research assistant and at Ege University, Turkey. So I have two pathways in my life. One is the academic pathway. The second one is the synthetic biology pathway and entrepreneurship pathway. And in my academic pathway, I did two master's degree, and I recently finished my PhD. And all of these projects are very different to each other. For example, I developed whole cell biosensors, and then I tried to characterize the multiple antibiotic resistance bacteria in the specific area in Turkey. And then I move on to the plant world with the investigation of the relationship between reactive carbonyl species and the endoplasmic reticulum stress in the model plant Arabidopsis thaliana. But in the other cases, in the entrepreneurship pathway, I'm an iGEMer, as all of the synthetic biology community knows the iGEM. And I start my iGEM journey in 2012 as an intern. And then I had a startup named Strand Biotechnology, which is supported by the National Scientific Council in Turkey. And then this year, I was in the judge in the iGEM competition, and I hope we will meet again in the next competitions as a different roles. And basically, I start with the synthetic biology in my life to developing biosensors, and then I move on to the nanotechnology to catch up the novel things. And last year, I developed the carbon quantum dots from the microalgae. And during my PhD, I focused on the plant stress physiology. And all of the crossroads of these three concepts is very excited for me because you can also understand from this actual picture. This picture shows us how the serious situation faced with us due to the global warming and the climate crisis. So between the help of the synthetic biology and nanotechnology, we can develop solutions for this kind of problems. Although when we talk about the climate change, many people think about the directly abiotic stress factors such as temperature, drought, pollution, salinity, etc. But plants face another stress factors like insects, which we call as a biotic stress. In both cases, these stresses are also have to be understand by the plants, and this is the first step of the stress physiology. If the plant understand the stress and perceive this stress, plant can be developed the responses which are directly related with the different part of the plants. For example, in the leaf, it can be related with the photosynthesis, which also related with the grain grain amounts in the wheat, for example, or in the roots, transportation or phytohormones regulations. And understanding these stress responses are very important for us because we have another technologies like visible NIR hyperstructural chip technologies or fluorescent spectrophotometers or RGB technologies or X-ray, CT, etc. And all of these instruments allows us to understand how can we improve the drawbacks of the climate crisis. And it changes our perception of the agriculture. For example, we can combine the navigation satellite or remote sensing satellite technologies with the help of the drones, and we can develop the uh, agricultural yield based on this situation. Similar to the Industrial Revolution, actually, this kind of opportunities change our perspective to agriculture. For example, with the drones or Internet of Things or software applications, we have to change ourselves as well. But in order to change our lives, we have to go back from the end user layer to perception layer, including the wireless network sensors or vehicles or robotic systems. In order to apply these ones, we have to first very well understand plant stress responses. So at that point, everyone knows that plants have some kind of responses, and reactive oxygen species has very important point of them, because when the plants perceive this stress, it creates some kind of metabolites, and this metabolites usually reactive species like reactive oxygen species, and all of these kind of reactive things has some outcomes like programmed cell death or systemic responses or membrane changes. And therefore, these kind of reactive species are studied very well. But nowadays, we know that subcellular ROS regeneration, intracellular ROS signature, even in the systemic approach, we can assume that the ROS are going over there, or we have some signaling pathways and etc. But we have to go deeper and deeper, and at that point, reactive carbonyl species are the novel players in the game, because we know that in the stress conditions, reactive oxygen species are produced, and this should be balanced in the redox homeostasis. But in some cases, oxidative damage cause some kind of oxidation in the lipids, and these lipids cause reactive carbonyl species, which has more longer uh, half-life than the reactive oxygen species, and it is related with the membrane integrity, and therefore they have some signaling function. In the plants, there are two routes to produce reactive carbonyl species. One is the enzymatic, the other is the non-enzymatic pathway. But the important one is the enzymatic pathway, because from as you can see from the here, according to the carbon number, there are lipooxygenases, and in the 13th carbon lipooxygenase activated enzyme cause some precursor of the jasmonic acid, which is a phytohormone, and it is related with the defense system of the plant. So if we observe this situation in the membranes, for example, if the plant stress and the lipids can be oxidized with the lipid peroxidation pathways, we observe the changes in the membrane permeability, which causes some kind of defects in the agriculture. And if we know all the pathway very, very closely, for example, if we know that the retrograde signaling pathway, which changes the gene expression, we can develop some kind of tools about that. For example, if we know how 13 of the AA change the turns in the jasmonic acid in the peroxisomes, and it is very important to deal with the stress. And as you can see from the figure, there are a lot of different reactive carbonyl species produced in different kind of compartments. For example, as I mentioned before, 12 of the AA is a precursor of the jasmonic acid, and it is very important to biotic stress. So if we know the pathway, we can deal with the biotic stress as well, because with the climate change, also the phloem feeders or chewing insects has some kind of differences, so we have to deal with them as well. Also, we have opportunity to use this knowledge with the benefit of the plants. So because redox homeostasis is based on the balance, it is very important if you produce some kind of reactive oxygen species, and then if you scavenge them and keep them in the balance, it could be help to the plant to be healthy. But if this balance is not occur, this time we face with the serious health problems in the plants. In agricultural point of view, it means a lot cost decrease. Therefore, as in the case of the reactive oxygen species, reactive carbonyl species also have to be scavenged. There are some kind of enzymes named aldehyde dehydrogenases, aldoketoreductases, or glutathione S-transferases, mainly important enzymes for the scavenging of the reactive carbonyl species. In that point, glutathione S-transferases are very important because in the plant cells, glutathione pool is very important and very key module of the redox homeostasis. And also some kind of importance has, as you can see from the figure, this balance is very important to glutamate, cysteine, and glycine metabolism as well. So therefore, if we know all this ratio, importance of this ratio, we can develop some kind of tools to improve our yields. Because glutathione pool, especially in the reduced form of the glutathione, has antioxidant and redox signal properties, or very important in the xenobiotic detoxification, or some sulfur-related pathways. They have very, very important roles. So therefore, we focus on the glutathione S-transferases, and we develop this kind of enzymes in some groups, especially named by the alphabet, alpha, beta, theta, and in among these tau groups in over here is very important to regulate the regulate the stress response. Therefore, we have some kind of question. Okay, we know that alpha, mu, phi, theta, zeta, and their 3D structures are very similar to that, to each other. But what about the tau class? And we can understand that from the literature. Tau class glutathione S-transferases are very, very important in the redox homeostasis because, as you can see from the here, these are very important two elements um, to balance all of the produced reactive oxygen species, and also polyunsaturated fatty acids are very important in the non-enzymatic pathways. So if we balance the reduced and oxidized glutathione amounts, we can definitely control all the stress response at that point. So what can we do with this knowledge? We can use this knowledge with the developing technology. For example, some kind of chips, as you can see from here, or single cell sequencing, epigenomic applications, genome editing, which I mentioned a little bit detail, and phenome, proteome, metabolome activities. And in agricultural perspective, we can combine these signals with the artificial intelligence to improve our agricultural perspective and combine with some problems that climate changing face with us. So at that point, the plant synthetic biology is, in my opinion, plant synthetic biology is the most powerful tool because we can manage many things, promoters, transcription factors, and we can build some kind of circuits, and we can use some knowledge from the ancestral plants to apply to genome editing perspective with the CRISPR and Cas systems, and some kind of metabolic engineering and heterologous plant systems to produce functional metabolites. So in that perspective, I want to give some examples. For example, Young et al. Shows us the novel synthetic inducible promoters controlling gene expression during the water deficit stress with the green tissue specificity in transgenic poplar. It is very important to develop some kind of promoters which is expressed in the green tissue in inducible form. And here they analyze the sequences and they create two different kind of synthetic promoters, and they express gas histochemical staining with them, and you can see that they also they found that in the green part of the plants they can induce the system with the water deficit, which allows us to use this kind of information as a water deficit sensors, for example. Another important example is a totally artificially designed synthetic promoter at that time, which the Bahaduria et al. Shows us. And they screen 2,500 different genes and their promoters with the two databases place, and the other database I forget it, but they use to create some kind of specific artificial promoter which has different kind of responsive elements, combination of the responsive elements, and they can use this promoter against salt stress. Here you can see the control group, and here you can see the 3 and 4, 150 millimolars of the sodium chloride application. And these are the transgenic Arabidopsis, and these are the non-transgenic ones. And you can see that they get a very good results. And why it is important to create some kind of stress-inducible promoters, we can develop many things like biosensors, like transgenic plants, and we can also use this information in the land to combine with the drone and the visual things to understand is the land is water deficit or not. And therefore, the application of synthetic biology is very broad. But the promoters are not only the source they can be used in the synthetic biology. For example, in the Niet cells and their colleagues' work, they create a fluorescent protein sensor based on the raw GFP, monitors in vivo hydrogen peroxide and the thiol redox integration in the cells. So they created some kind of protein couples, and based on the sulfur groups over there and reduced state oxidate state difference says there's a redox difference and there's a hydrogen peroxide uh, production, which means there's a stress over there or not. And we can see from that this production of the oxidized state proteins also related with the phenotype. And also we can use this again as a marker for the stress. But interestingly, today I found very interesting paper which Dotan et al. Shows us. They also use this knowledge to create electrochemical sensors and use this redox cycling perspective. But in that time, they create some kind of different sensors to put the stem of the cells, stem of the plants, and they use these redox reactions to understand if there's a stress or not. So we can directly say that if we know how these molecular methods are used, we can also develop this kind of sensors as well. But story is not on here, and therefore we have to mention about metabolic routing strategies that enhance the resilience and the production. As far as I know, everyone hears the golden rice. It's a very old but gold thing. And also shows us we can save millions of lives with the help of this plant synthetic biology. In this golden rice case, you know that there is a some kind of genomic, transcriptomic, and proteomic approach and integration of the precursor of the vitamin A to the rice. And therefore, although it is a very, very old technology and old paper, it's rooted from the very different perspectives like Sanger sequencing, microarrays, 2D gels, and PCR, and whole genome sequencing, differential expression profiling. But all of the stuff has a very, very good baseline for us because today we have many, many good tools to improve our crops and also improve our perspective to improve our lives with terms of the agriculture. For example, there's a genome editing tool that many scientists in the many scientists in the world use the CRISPR, and we can use the knowledge based on the model plant species or traditional plant species and use the bioinformatic tools such as NCBI and other databases. We identify many stress-related genes or promoters or some kind of mechanisms, for example, glutathione S-transferases, and we can integrate this knowledge to novel plants and agro-infiltration and transformation technologies, and we develop some kind of resistant uh, plants, which enable us to improve our agriculture. For example, in here you can see that Lemon and colleagues are use this technology to improve the CLV1 gene to improve the fruit size in the plants. So they combine the domesticated mainstream crop to gene edited version, and this also saves the many nutritional values. So therefore, we can reach nutritional and agricultural perspective very better options because we need to do that. As a future perspective, I can directly say is that we have very good tools and we have very good molecular pathways that resides in the cells of the plant. So okay, we know that world is changing, we face with the climate change, we have global warming, and everything is changed, including the stress factors such as biotic stress factors or abiotic stress factors, and we have to adapt our agricultural practices according to do that. But when we do that, we also have to be careful about the food security part, and it is not enough to limit the chemicals that we use as a fertilizers, or we try to save the microfauna, microorganisms, or fungi, or any physical things that we use in the land. It's not enough. So we have to move on in terms of the synthetic biology and the cases that we have, novel things that we have. So I hope we will do this as soon as possible and we reach secure and good food with the agriculture 4.0 applications. And thank you for your participation. And if you have any questions, I can answer them. Thank you.
Thank you. If you guys have any questions, you can drop them in the chat. I have a question, and we can go to Davide. I saw you had a lot of different examples from Metabopsis, and it was very cool to see that even trees are being engineered. Yeah, because you never imagine genetic engineering to happen with trees, right? Why
Yeah.
would you engineer poplar
Actually,
out of
I
all the trees?
choose that example because poplar use as a model organism at that they claim that they use poplar because of the they claim that model organism for many different kind of applications. So we know that okay, Arabidopsis is a model organism and it is glycophytic and okay, but it has very relatives and it is very hard to modify the trees actually. So they prefer to use the poplar at that point. So I just want to mention about this kind of different application because we
Mhm.
don't limit ourselves with the Arabidopsis or Lemna, which is a very easy to transform, but we have other options as well. I just want to mention about that.
Hmm.
But it is
And
very hard to genetically modify the tree or whole forest, for example. But at that case, as far as I know from the literature, they usually use the fungi because all the trees are communicated with the fungi, and they modify the fungi and they apply the modified fungi to communicate the trees at that point, as far as I
oh,
know.
like a vector for the
Yeah, yeah,
genetic
kinda.
engineering. Oh,
Yeah, kinda.
oh wow, that's very cool. I wonder what kind of like security aspects you would have, for kill switches and things like this.
Yeah, yeah, yeah. We
But
should have to do that because
yeah,
uh, as I mentioned, okay, global warming and the climate crisis are very hard things and very important things, and we have to be faced with that in very, very close time. But we also have to respect nature at that point. So we have to limit our things with the kill switches, maybe with the another things, and we have to be careful about that. Biosecurity is also another point of this issue.
yeah, and I think plants are very important because they were a bit of the drive of the like anti-GMO movement, right? And
Yeah,
with the golden rice now, like most countries don't accept it, even though it is a good thing.
yeah, yeah.
Um,
We have
so
to develop this kind of actually awareness. All the GMOs are not bad.
yeah,
Okay, it can be bad. I also can say that because the genetic modification is very how can I say that, very very strict. It should be very strict. But
yeah.
if somebody needs some kind of molecule and you can do this with the GMOs, including, for example, the bacterial production, you also produce some kind of drug components with the bacteria as well, for example, insulin. So it is the same thing actually. And we have to be raise this awareness, I guess.
It is interesting though because people take medicine from GMOs all the time and they don't realize it. But the moment they're eating it in a tomato that has more vitamins or is better at drought resistant.
Also, with this kind of technology, actually, we have some kind of opportunity to, for example, create edible vaccines as well. It is also another topic for the plant science, but actually, I also think that I had a very agricultural perspective actually. So I try to a little bit move on the agricultural part.
Mhm. Okay. Davide, do you have any questions?
Yes, yes. Thank you for the very nice presentation. And yeah, I just want to say first of all, I fully agree. We have to repair some of the damage that ourselves have caused, but we should be aware that we should also limit the damage that we are making. I wanted to ask you, in the context of possibly scaling up the solution to solve some of the problems that our crops have and will have more and more in the future. Do you know if the enhanced response to stress could impact the yield or growth of the organisms?
Actually, as far as I know, especially for the biotic stress factors, it is very important to deal with them because if one plant get infection, it is very easy to spread. So as far as I know correctly, I'm not sure, but I need to check it. They use some kind of drones on the lands, and they take pictures of the lens like a pixels, and they use this information to compare which plant is diseased or not. But they use this technology for biotic stress. But I think with the help of the abiotic stress sensors, it could be useful for another stress factors as well.
Okay. And as another question, maybe even more general, how do you think that, for example, there could be countless combinations of different backgrounds for the organisms going from different soils to weather conditions, yeah, anything. Do you think there could be a general solution that might solve a big subset of these conditions, or each problem would require a specific tailoring? Like, how general is this solution that you have just presented?
Uh, Actually, the main aim of these studies are understanding first individually by the stress factors. For example, heat stress, cold stress, or drought. That's okay. But in the cell or in the plant organism, they face all of the stressor at the same time. Therefore, we have to combine stress factors. We have to understand the combination of the stress factors. Therefore, we a little bit move on there, but we have a lot of way to do that. For example, biotic and abiotic stress factors are used very different pathways. So, for example, cold and drought could be similar pathways. Okay, they share many things, but for example, heat and freezing has no common thing. So we have to, in order to find this kind of general solution, we have to very be precise about all the pathways uh, that this stressor cause. So we have to first understand the stressor alone, and then we start to combine them to each other. So as far as I know from the literature, the combination study is also very hot topic right now. I hope one day we will find the one general solution, but it is very a little bit far away.
Yeah, it would be already good to find some good combinations, let's say, that could work, and
Yeah,
apply them
it
to
requires a
regular.
lot of optimization. For example, we always talk about if the 1 degree Celsius change the climate, how it is affect. We have models and we have a lot of things, but this Celsius degrees change could, for example, block some enzymes. It is very important. So therefore, everything is changed. One enzyme could be non-useful, and another enzyme could uh, finish its work. And cell is very crazy about that. And plants has a lot of things like that. They are incredible creators.
Yeah.
I have another question. So I work with plants as well. And I've worked a bit with trees, willows, but also with tobacco plants. And my question to you is, what do you think is stopping plant science from having the same hype as the fungi? You know, like we see a lot of like sustainable meats made with these, but nobody's getting the hype of talking about plants. Even though there's much more vegan people, like the rise of vegan people is increasing in the world. What do you think is stopping plants for having this some kind of attention?
Actually, I think plants needs more attention. I know that, but it is very hard, for example, to grow a plant in the laboratories when we compare to bacteria or fungi. And therefore, scientists more prone to use them because they need to be quick and they don't want to wait, for example, 1 week to germination. Or, for example, in the tree case, it takes almost years and it is very hard to sustain this kind of things. But I think one day all the researchers are understand how plants are incredible because as a population in the world, we have to fed up. We have a lot of people around us, and they all need food, and they have to process the food, and we have lands to use, and also some of the world has some kind of extraterrestrial things in order to go to there and colonize that place, and we have to plant something, and we have a lot of way actually to understand how incredible the plants.
Yeah, yeah. And they're a great example of, like, again, we just need light and water. While all these fungi, they might need this complex sugar mixes to grow and things like this. So,
Also,
yeah.
we have a lot of extremophile plants as well.
Mm-hmm.
Yeah,
Yeah.
that's
But they
so.
don't get, but it's all Arabidopsis, isn't it?
Yeah, we all talk about Arabidopsis, okay, but we have a lot of things uh, living in the extreme conditions.
Yeah, yeah. And they're not explored much because it
Yeah,
might take quite a few years to understand the
a
physiology.
lot of huge
Yeah,
world.
yeah. Well, if there's no questions from the public, we can finish it off here. Next year we're gonna have a Plants in Bio Festival in September. So if you liked this talk, yes, you're going to enjoy that festival even more.
Oh, I will want to do that.
Yeah, yeah.
I hope
We're still in planning stages, so.
I hope at that time we will meet in person.
Oh, yeah, yeah. Yeah, so thank you. Have a great Christmas and a happy New Year.
Thank.
We'll come back in January for the next seminar.
I wish you, all of you, to very Merry Christmas and Happy New
Thank
Year
you.
as well. I hope you enjoy your Christmas.
Thank you. Thank you.
Nice to meet you.